How can cell cycles switch from mitosis to meiosis? This is a very fundamental question in biology, but we still don't know much about the mechanisms how meiosis is initiated. Before meiosis, germ cells are growing under mitotic cell cycles. Then germ cells spontaneously undergo meiosis in response to stimulation by retinoic acid. Upon the stimulation by retinoic acid, germ cells transiently express STRA8, then enter into meiosis. STRA8 was originally identified as a cytoplasmic factor, which is induced by retinoic acid, but there is no canonical domain in STRA8 protein. Another study showed that STRA8 knockout germ cells do not undergo normal meiosis. These studies suggest that STRA8 should play an essential role in meiosis. However, the precise mechanism of meiotic initiation was still not clear. To find the answer, we attempt to purify the meiotic factor STRA8 using a knockin mouse line. Using the knockin mouse line, STRA8 protein was purified from the testis. Unexpectedly, by mass spectrometry analysis, a new protein was identified, which was encoded by a hypothetical gene, GM4969. And we named this newly identified protein the meiosis initiator, meiosin. Meiosin homologs are conserved in vertebrates and encodes helix-loop helix and HMG-like domain, suggesting that meiosin may be a DNA binding factor. Next, we examine the expression pattern of meiosin in different tissues. Meiosin was expressed in testis and embryonic ovary, suggesting that it is a germ cell-specific factor. In testis tubulis, meiosis progressed from the outer to inner. Here, SYCP3 indicates spermatocytes in the progress of meiosis. Meiosin was expressed in pre-meiotic cells, but not in SYCP3 positive spermatocytes or haploid cells. And meiosin was co-expressed with STRA8 along the outer basement. Altogether, these indicate meiosin protein is expressed before the initiation of meiosis in testis. Also, in embryonic ovary, meiosin was co-expressed with STRA8. As I explained earlier, STRA8 is expressed just before meiosis initiation at the transition time from mitosis to meiosis. So this means meiosin is expressed at the transition period from mitosis to meiosis in the ovary. Next, we generated meiosin knockout by CRISPR-Cas9. In meiosin knockout, testis was smaller compared to wild type. In the knockout testis, sperm was absent, which means the knockout male is infertile. By immunostaining, although there are SYCP3 meiotic cells and haploid spermatids in wild type, in the knockout, STRA8 was expressed, but SYCP3 positive meiotic cells and haploid cells were absent. So the initiation of meiosis failed in meiosis knockout. Here's another data. In wild type, homolog chromosomes undergo homolog pairing and recombination to form bivalent chromosomes during meiosis 1. SYCP1 is a marker of homolog pairing. As you see, the knockout cells did not show any sign of homolog synapses. This indicates that meiosin knockout cells failed to undergo homolog synapses and meiotic recombination. Then, without entering meiosis, what happened to meiosin knockout? The question here is whether meiosin knockout cells are arrested or still growing. Histone H3 serine 10 phosphorylation is a marker of mitotic chromosomes. We notice that meiosin knockout testis show high accumulation of mitotic-like cells indicated by histone H3 serine 10 phosphorylation. This shows the phenotype of adult ovary. In meiosin knockouts, the ovary is degenerated and the size is smaller compared to wild type. But a small number of oocyte-like cells are still found in the knockout. By Gibbs staining, we can see 20 pairs of bivalent chromosomes in wild type, each pair bridged with chiasmata. In contrast, in meiosin knockouts, we see 40 sets of univalent chromosomes or 40 single chromatids lacking chiasmata. This suggests that the oocyte-like cells in meiosin knockouts did not undergo meiotic recombination, which means without meiosin, meiosis cannot proceed. Next, we analyze the transcriptome in meiosin knockout testis. This scatter plot shows the gene expression profile of meiosin knockout versus wild type. These green plots are the downregulated genes in meiosin knockouts. By gene ontology analysis, we can see a significant number of meiotic genes are downregulated in meiosin knockout. This accounts for the failure of meiotic entry in meiosin knockouts, as shown in previous slides. Because meiosin has a putative DNA binding domain, we examine the chromatin binding of meiosin and STRA8 by ChIP-seq. As you see here, meiosin and STRA8 bind to the promoter regions of these meiotic genes. Moreover, by Kei-ichiro that indicates transcription start sites, we can see that meiosin and STRA8 do bind to the transcription start site of these meiotic genes. From the transcriptome analysis shown in the previous slide, together with these chromatin binding analysis, we can see that MEIOSIN-STRAS complex regulates the transcription of meiotic genes. In summary, the MEIOSIN-STRAS complex is required for cell cycle switching from mitosis to meiosis. The MEIOSIN-STRAS complex activates meiotic gene promoters. That's all for now, and we would like to thank all the collaborators. Thank you for your attention.